Hi. So in previous session you have seen various plagues or disease, and we people learned how to protect from these infection. So after that, the scientific community or those who are interested in understanding the basis of this protection, they further moved, and they have found out there are several school of thoughts. And so in this session basically we will see how this immunology, basic immunology field was initiated, established, and thereafter we will also discuss about the all those great works which received the Nobel Prize, maybe in this session or maybe next session. Okay. So there was a dichotomous thinking in immunology. Dichotomous means there are two opposite or quite polarized thought was there, that the defense whatever we have or the animal have, some people thought that or some people say that this is due to some cellular component, some cell. Okay. And another school of thought is that no, there is some factor which is present in blood and that is giving a protection. So this we call it as a dichotomous thinking, and both school of thought they are very against each other. Okay, it's not that they don't want to compromise at any cost. So I will take you through those two school of thought, and now you are also maybe aware that both theories were correct. It's the defense is not only by some cells, but it is also there are some soluble factors, okay, which is also playing a very important role in defense against any infection. So let's look at this what is this dichotomous thinking, who are the main proposers and who are the supporters for these two school of thoughts. Okay. So here you can, I have a one very nice slide which I have taken from this article. If you want to see this article, then you can take this article and go through. Anyway, I am going to talk about what are the two school of thought. So one school of thought is, we call it as a cellularist. Cellularist means all those scientists who believe that there are some cellular component. Okay, and this theory was not theory, this observation was given by Emil Metchnikoff. Here you can see there is a on top Emil Metchnikoff. He gave this theory after observing some of his work. Okay. So basically his theory was that the chief or the key defense in the individual or in animal is by some specialized cell which eat the pathogen. Okay, and they call these cells as a phagocytic cells. Okay, and these phagocytic cells digestive property imparts the immunity in the host. Okay, he also said that there are some macrophage. Macrophage is basically polymorphonuclear leukocytes. These are the type of blood cell which I will discuss in great length when I will take the cells of immunity. Okay. So basically this macrophage is playing a key role in immunity, and their digestive capacity or digestive power gives the strong immune response. Okay. And who all are the supporter of this theory? One is the Charles Darwin, who was saying that okay, this phagocytosis as a general mechanism of host defense. Okay, he was one of the supporter. Another is that Rudolf Virchow. He also he also supported this theory by saying that cells are the central in immunity, in defense. Okay. There is a Julius Cohen. He also supported this theory, who discovered white blood cells which is present at the site of inflammation. So, we will discuss all these white blood cells and inflammation in great length in future sessions of this course. On another hand, there are another school of thought, we call it as a humoral realist, and these basically, the humoral means the soluble component. Okay, the soluble component which is present in body fluid, mainly blood. Okay, this school of thought was basically supported or initiated by Paul Eckerlich, and this person is basically showed that okay, there is some soluble substance which is present in the blood and other body fluid. They are basically destroying the invasive pathogen. So, this was his concept, and there are several supporter of his concept. One is that you can see that Emil Fischer, who basically gave this concept. So, there is a pathogen and there is some unique molecule which fit with this pathogen like a lock and key. There is a unique molecule, okay, and this unique combination basically eradicate the pathogen from the host. Okay. Another is Emil Behring. So, he basically gave this concept of antigen. Antigen is any substance which is foreign to the host. Okay, antigen, and he gave the principle of antigen antibody interaction or reaction, and he was also one of very strong supporter of this theory. Another is Julie Bordet. So, Julie Bordet also a very strong supporter of this theory, and his work also resulted to support this school of thought, that there is some soluble component which interact with pathogen and then that will kill the pathogen. Okay. So, let us look at about the work of Metchnikoff and Eckerlich. Okay. So, phagocytic theory of immunity basically given by the Metchnikoff around 1884. Okay. And he observed that when the starfish, he introduced some thorn in his starfish larvae, and the amoeboid cellular entity that seems to be engulfed the thorn. So, he observed that there are some amoeboid kind of cell which is taking up this thorn and basically digesting this thorn. Okay. And Metchnikoff gave a term of phagocytes, the cell which eats, he called it as a phagocytes. And then his theory got a lot of support, okay, because he demonstrated that there are some cell which eat the pathogen or eat the foreign thing. So, this gives the concept of cellular immunity. And Metchnikoff basically hypothesized that the cells rather than the soluble component is playing an important role in defense. So, he was against that all those school of thought which is supporting that there is some soluble component and giving a defense. So, in that way Metchnikoff work, he also received the Nobel Prize for this this work. Okay. Another is Paul work. Basically, he was a physician, and he is more interested in chemistry component. Okay, and he also gave one theory, which is, we call it as a side chain theory. Okay, so basically, when there is a toxin in the host, and then there will be some molecule will be generated over the cell, okay, which he call it as a side chain. Okay, this side chain is basically a cell protoplast, which is basically antitoxin in nature, and he also call it as a heptophore. Okay, this heptophore, because there is no clear word about like a antibody. Okay, so initially he call it as an antitoxin. So the molecule which is generated over the cell, and this molecule is binding with the say the foreign substance or toxin, he called it as a heptophore, and this molecule basically neutralizes this the foreign substance or pathogen or molecule. Okay, so cell under threat of foreign microorganisms grow this extra side chain to capture the toxin or to capture the microbe. Okay, so he gave, so you can understand at that time the field was just beginning, the immunology field was just beginning, and you can understand this is what kind of thinking people had. There is some molecule which binds, and basically this captures the antigen or toxin, and then basically it neutralizes. And you can think that in 1897 this was quite correct. Now there is a B cells, they express the antibody molecule. Okay, and then these B cells basically differentiate to the plasma cell and produce antibody. Okay, so these additional side chain basically designed to break. So the cell is making this side chain, and when this side chain will bind with antigen, then this will break, and then this will present in the circulation or in the blood. Okay, and this is quite very or very well supported. This theory was very well supported by many workers. Okay, so the first model of to propose for an antibody molecule in which the antibody was branched and consists of multiple site of binding to foreign material known as antigen. Now we know, we call it as antigen, and for the activation of complement pathway. Okay, so this was now we know that this molecule is expressed, and then this is binding with the antigen and eliminating, and in addition this is also used for the activation of complement. Okay, so these were another school of thought, and by this work there was a two school of thought, and there are several people who supported this thing because in their observation they found out similar things. So now I will talk about the isolation of diphtheria toxin, which is basically given by the Emil Rox and Alexander Yasin. So basically they have collected the bacteria, diphtheria bacteria from Kleb and Loffer. Okay, and they observed that this bacteria is not causing the illness, rather some toxin produced by this bacteria is responsible for causing disease. Okay, and his work was quite well accepted by at that time immunologists, and now we know that diphtheria toxin plays a very important role in pathogenesis. Thereafter there was other discoveries, like people showed that there is some factor present in the blood, and this factor is having a bacteriolysis property or bactericidal property. Okay, So this work was basically initiated by George Nuttall, and he basically provided that there is some factor which is present in a fluid or body fluid, and this basically kills the anthrax bacilli. Okay, and when we take this blood and heat it to 55 degrees centigrade, then this capacity is disappeared. Okay, and if we heat this blood at 55 degree, but again if you replenish the reaction with fresh blood, okay, or fresh blood serum, then this killing capacity is again the bacteria will be killed. Okay, again regain this killing capacity. Okay, so here you can see that the concept of complement is emerging, okay, by his work. And basically there is another supporter who Edward Buchner, he called this factor as a alixin, and which is basically destroying the bacteria. Okay, so there was one more scientist, his name is Richard Pfeffer. So he basically showed that in animal who received this Vibrio and cleared this infection, if you inoculate again the bacteria, then this bacteria will disappear from this animal. Okay, and he also demonstrated the similar property in vitro condition. He have taken the fluid and then he put this bacteria, and then there will be a lysis of bacteria. Okay, and basically this observation got a lot of recognition, and people recognize this phenomena as a Pfeffer's phenomenon. Okay, and this is basically used for differentiating closely related pathogen. Okay, so finally all this theory was well explained by this Julie Bordet, who was the discoverer of the complement, and he showed that, okay, when the blood serum is heated at 55 degrees centigrade, then this protein, or there is a group of protein, later on it was called as a complement, was basically losing its property, and this can be replenished if we provide the fresh blood serum. Okay, so the complement word is basically a set given for the family of protein. Okay, and we will discuss a lot about the complement. We will study the pathways of complement. So why complement word is there for this family of protein? So here just I would like to tell that this complement basically complements the blood property. So if you have a little knowledge, this complement is also activated by antibodies. Okay, and so when there will be a lysis of bacteria, and if you put the complements, it complements this lysis property. So that's why this complement name came. Okay, now there is a serum as a protection matrix. Basically this is a work of Emil Behring, and he basically gave the concept of prophylactic serum therapy, and for this work he received the Nobel Prize. Okay, and at that time this prophylactic serum therapy is considered as there is some antitoxin, okay, which is giving the protection. Okay, and this is one of his very beautiful experiment. Here I would like to explain this experiment. Here you can see that there are three groups of guinea pig. Here you are seeing only one guinea pig. Okay, and the first guinea pig did not receive any toxin, and the second group they received the toxin from strain A, okay. And please remember, this is a sublethal dose, not very lethal dose, okay. And the third group of guinea pig received the toxin from strain B, okay. After that, what they have done, they have isolated the serum from these challenged guinea pigs, and then they introduced the serum in another group of guinea pig, okay. The first group, here you can see that the serum is from uninfected or unexposed animal, okay. In second group, you can see that the serum is taken from those group which receive the sublethal dose of toxin A, and in third group you can see that the serum is taken from the animals which is challenged by strain B toxins, okay. And then after that, they have infected all these three groups of guinea pig with this strain of challenged by basically the lethal dose of this toxin A, and then they have observed that the guinea pig which received earlier received the serum from the animal which is which are challenged by the sublethal dose of toxin A, got protected but not other. So this shows that this is quite specific, the response is very specific, okay. So, and this response is basically present in the blood because they have used the serum, okay. Thereafter, there was a other work in 1896 in which they have showed this agglutination of bacteria by Max von Gruber and Herbert Durham. So basically, they showed that bacteria of similar size clamped together in sera in ways specific to or determined by each serum, okay. So basically, agglutination assays and all those things came in picture, and one very good example of this agglutination assay is the Vidal test, which is given by George Fernand Isoder Vidal, and this Vidal test we use use for this typhoid, okay. So here I gave you kind of a middle age overview of immunology, okay, and now towards end I will discuss about the or in next session I will discuss about all those work which received the Nobel Prize, and then finally I will discuss about the different branches of immunology. Thank you very much.